Hello everyone. In this presentation I will be talking about protein synthesis regulation. So before I get into protein synthesis regulation, I wanted to take a moment to talk about the central dogma of biology and protein synthesis. So the central dogma of biology basically is regarding the information flow from DNA all the way to protein, so to speak. And in a nutshell, basically the human body has DNA and that is transcribed into messenger RNA, and that messenger RNA is then translated into polypeptide or protein, um, and that's how the DNA sequence is transcribed first and then translated. Now, in terms of endocrine regulation or hormone signal regulation, we have um, the slow regulation and then fast regulation. This slow regulation usually is actually transcriptional, not usually. Um, and with the slow regulation, it involves a protein called GCN2, general control non-derepressible 2 protein. And this protein basically senses the deficiency in amino acids, and what it does is it decreases protein synthesis and increases amino acid biogenesis and transport. And we'll get more into detail regarding these last two bullets in the next slide. For the fast regulation, it typically involves a protein kinase, and there are several hormones that activate mTOR. So the fast pathway is mediated through mTOR, and there are a group of hormones, which we'll mention later in this presentation, that activate mTOR. And there's also the glucocorticoid cortisol that reduces mTOR activity. Now, before we get into, I said we will be talking about the last two bullet points here of GCN2 regulation. But before we do that, I wanted us to take a moment and talk about this eukaryotic initiation factor 2 alpha. Now, this initiation factor, it is required in most eukaryotic cells to initiate translation. So it's this step right here, and it mediates the binding of the tRNA, which is charged with an amino acid, to the ribosome in a GTP-dependent manner or energy-dependent manner. Now, this initiation factor, when it's phosphorylated, it does not translate that messenger RNA. So the messenger RNA is not translated to a polypeptide. So looking at a little bit more closely into the GCN2 activity, we said that it decreases protein synthesis, and it does that by phosphorylating and inhibiting the initiation factor EIF2 alpha, and therefore inhibiting protein synthesis. And it increases amino acid biogenesis and transportation by activating a transcription factor called ATF4. And this increases the enzyme levels involved in amino acid synthesis and transport. And I just wanted to here represent these, where these two, where does this transcription factor fit into the central dogma, and where does this initiation factor fit in? So the ATF4, it inhibits transcription when it activates transcription. So when this is inactive, transcription will be inactive. And same with EIF2 alpha. When it is active, we have active translation. When it's inactive, by phosphorylation, we don't get any translation of messenger RNA to protein. Now, GCN2 mediates the fibroblast growth factor 21 production and the FGF21. And FGF21 is a liver-derived hormone that rises in response to low protein. So far it's unknown how does FGF21 restore protein homeostasis, but it is thought that it happens by increasing appetite, because the only way to increase non-essential amino acid levels is by actually consuming them. So just to take a closer look at the fast regulation of protein synthesis, we said it's mediated by mTORC1. So the hormones that activate mTORC1 is IFG1, which is insulin-like growth hormone, and then we have insulin and testosterone. The reducing mTORC activity reduces protein synthesis, and the glucocorticoid cortisol does that, reduces the mTORC1 activity. And mTORC1, what it does, it promotes protein synthesis by promoting messenger RNA translation, ribosomal biogenesis, and suppressing protein breakdown. And this is my presentation.